All right, when we talk about cells carrying genetic material, it was a while back ago that actually, in thinking about it, geneticists like Mendel wondered, when a cell copies and it has this genetic information in it, specifically what location in the cell would this material be located? Okay, and we started wondering that, which were the structures inside of the cell that would carry this information and pass it on to the next cell. And obviously our answer to that is in the what? In the nucleus. Okay? Now we've always seen the nucleus as a ball type structure. But in reality, that ball type structure is actually a bunch of set of strings or string-like structures that are our chromosomes. And they get all balled up exactly like a ball of yarn, and it makes that nucleus. And inside there, we have what we call our genes. Think of genes as being a section in chromosomes. So if you took a chromosome and you kind of straightened it out, if you would, kind of like draw something like that, and then this little piece from here to here, just this little section right there, that's what we call a gene. The whole entire thing would be a chromosome, but that little section right there is what we refer to as a gene. Now, when talking about genes, you have to remember that in one particular gene, you're going to have information that's important to whatever traits you pass on to your offspring, whatever traits were passed on to you. And also it's important to remember that no one gene is responsible for an entire trait by itself. Something like, for example, eye color is not going to be determined by one gene. It's actually multiple genes put together that are going to consist of what color eyes you're going to be born with, what color hair, what texture hair, all those characteristics that you inherit from someone and also pass on to your offspring. And they don't all come from one chromosome either. They can come from multiple chromosomes. So it could be a section on this chromosome together with a specific section on the same chromosome together with another section off of another chromosome, and the three put together is what could make up, say for example, the color of your skin, okay, or any inherited characteristic that you're going to have. Now, how many sets of genes would you have? Well, think about it. If you got some inherited characteristics from mom and some from dad, even though only one shows, you still got what? 2 sets. You have 2 sets of inherited characteristics, or 2 sets. For every gene that you have, you're going to have 2 sets. One from mother and one from your father. Now, there are 2 terms that you have to learn here. One of them is going to be diploid, and the other one is going to be haploid. It's very simple to understand. A diploid cell is what we typically call a body cell. It's another way of referring to a diploid cell as a body cell. So the cells that make up your blood, skin, eyes, brain, bones, all those cells, even the specialized cells that make up your kidneys, heart, lungs, and so forth, those are diploid cells. Di means 2 sets, right? You could remember it as body cell, but on the EOC it's probably going to say diploid cells. They typically don't use the term body cells. I'm giving you the term body cell that you can understand it as all the cells that comprise in your body, in your bones, in your organs, your blood, and so forth. Di means both sets. That means that that one cell, the cell in your skin, the cell in your bones, the cell in your brains, your blood cells, all those cells, those diploid cells, contain both sets. A set of chromosomes from your mother, a set of chromosomes from your father. How many do we have? We have 46 chromosomes in total. 23 from mom, 23 from dad. So we say that we have 46 chromosomes. We also refer to it as having 23 pairs. Okay, 23 pairs. 2 number 1s, 2 number 2s, 2 number 3s, all the way up to 23. One from your mother, one from your father. In a cell that has both sets, we refer to that as a diploid cell. But you have one cell in particular that only has one set, and that would be a haploid cell, HA, and the word half also starts with the letters HA. So a haploid cell is going to be half of those chromosomes. It's not going to be one from mom and one from dad. It's just going to be one chromosome number 1, one chromosome number 2, one chromosome number 3, and so forth. And there's only one cell that you have in your body that we refer to that as. Those are what we call your sex cells, your reproductive cells. In a male, it is what? Sperm. And in a female, it is the egg. Those are the only haploid cells you have. Males only have one haploid cell, sperm. Females only have one haploid cell, eggs. That's it. Every other type of cell is referred to as a diploid cell. Remember that the nucleus inside of these cells is made up of those same chromosomes that we're talking about. Now in that chromosome, or in those chromosomes are what we refer to as the strands of DNA and proteins that when they all bundle up, they make that nucleus. And those are where our genes are going to be found. Now this picture shows a picture of an adult fruit fly. An adult fruit fly has 8 chromosomes. Here we have the 2 green ones, the 2 red ones, the 2 yellow ones, the 2 dot-looking ones. Okay, those are the 8 chromosomes in a fruit fly. See how they're paired up? The 2 reds look the same. The 2 greens look the same. We refer to that as homologous pairs. There's that word right there, homologous pairs. It means that each of those 4 chromosomes, the 4 that it got from its mother and the 4 that it got from its father, are going to be the same. And in this process, which I'll explain in detail later, those pairs, those homologous pairs, are actually going to find each other. And number 1 and number 1 are going to pair up and stay together. 2 is going to find the other 2 and pair up. 3 and 3 and 4 and 4 are going to find each other as well. And they're going to make a homologous pair. The adult fruit fly has 8 chromosomes in total, 4 homologous pairs. Okay, each set that it gets, one from its mother, one from its father, one from each parent. Now, that means that when that one cell has everything put together, that's a diploid cell. And this diploid cell will eventually split up. And this red will go into one side, this red will go into the other side. One of the greens will go into one side, one of the greens will go into the other side. And so that diploid cell is going to do what? It's going to split up and it's going to make haploid cells. It's a lengthier process, but we'll get there. Now, in a diploid cell, we refer to that as having a symbol, the diploid cell having 8 chromosomes, as 2n. 2 of whatever number of chromosomes exist in the haploid cell. So in the case of the fruit fly, 2n is going to equal 8. Okay? In us, 2n is going to equal 46. Okay? So our diploid cells, which are going to be, think of 2n as another form of just referring to as a diploid cell. When you see 2n, you know you're talking about a diploid cell because it's the number of chromosomes times 2. You're rarely going to see that, and there's a good possibility you won't even see 2n on the EOC. However, the chance is there, and I want you to be exposed to that. So, some of our cells, our haploid cells, contain only a single set of our chromosomes. So those cells we refer to as haploid cells. That prefix HA, excuse me, means one set. That's also referred to as a sex cell, reproductive cells, which in the end, when we're talking about either a sperm or an egg, we refer to those as our gametes. Okay, the gametes of the sexually reproducing organism are haploid. Sperm are gametes, eggs are gametes, they are haploid cells. So if our diploid is referred to as 2n, our haploid is simply referred to as n. So now we get into the phases of meiosis. Meiosis and mitosis are very similar. Mitosis can be seen in kind of in a way as half of meiosis. The first half specifically, with a slight change. But if you understand meiosis, then mitosis is that much simpler to understand. So I'm going to go through the phases of meiosis, which are obviously much more complicated than mitosis. So what happens during each phase of meiosis? Later we'll get into representations with illustrations, and when you see those visuals, you'll get to understand it a little better. But in a nutshell, first I want you to, I know it's here on the board, but I'm going to draw it here. I want you to memorize this. That's an A, by the way. All right, PMAT. If you memorize PMAT, you've already got the order for the process, for the phases of meiosis. The P stands for prophase. Okay? And the M stands for metaphase. On the next slide, we see the next two. A stands for anaphase, and T stands for telophase. Now, PMAT is also obviously something relatively simple to memorize. MAT, 3-letter word, you learned that when you were about 3 years old, and just put a P in front of it. PMAT, prophase, metaphase, anaphase, telophase. Now, in prophase, let me bring this back. In prophase, no, let me clear that. In prophase, each chromosome is going to have a replicated pair. Now, replication just basically means that that chromosome is going to split, not completely, but think of it as like a hot dog, if you will. And if I draw this out, I think it'll be a little easier to understand. Think of the chromosome as being something that kind of looks like this. Now imagine if I took a knife and I cut right here, and then I did it again here. And I just basically cut that way, leaving the middle connected or intact. What you're going to end up with is you're going to end up with something that looks like this. And the same thing on the other side. Okay? My cut ended up being right here and right here. So you end up with this. Okay? That's the first replication that occurs in meiosis, where the chromosome simply goes through this cut, and you end up with something that looks like this. Now, in prophase 1, and again, before I continue, let me kind of explain this. Meiosis has two phases. It has a meiosis 1, and it has at the end of 1, we go through meiosis 2. Now, meiosis 1 is going to go through that same PMAP. Prophase 1, metaphase 1, anaphase 1, and telophase 1. At the end of that, it's going to go through basically with a slight difference, but basically the same thing all over again, but then we refer to that as meiosis 2. And it would be prophase 2, metaphase 2, anaphase 2, and telophase 2. And by then, actually, then we're done. Okay? So it's going to go through the process of meiosis 1, which I'm going to explain now briefly, and then at the very end of that, it's going to go through the same thing, kind of, all over again, but we refer to it as meiosis 2. And we would refer to as a prophase 1, then followed by metaphase 1, then anaphase 1, then telophase 1, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. So those 8, one after the other, the whole entire thing ends up making meiosis. Meiosis is how a sex cell reproduces or forms. It is basically turning one diploid cell into 4 haploid cells. Okay, one body cell into 4 reproductive cells. Mitosis is just basically a bone cell making another bone cell, or a blood cell making another blood cell, a brain cell making another brain cell. Okay, so mitosis is just basically half of it. But meiosis is much more complicated, because here's where we're talking about the separation of these chromosomes to be able to get there so that you can produce fertile offspring. All right, in prophase 1 of meiosis, this is what occurs. All right, let me bring this back to here, clear all this up. So, in prophase 1 of meiosis, each replicated chromosome is going to correspond or find its homologous chromosome. Remember that we're first starting out with a diploid cell. So there are two number ones, 2 chromosome number 2s, 2 chromosome number 3s. In the very first prophase 1, those replicated chromosome pairs are going to pair up with its corresponding homologous chromosome. 1 and 1 pair up, 2 and 2 pair up, 3 and 3 pair up, 4 and 4 pair up. Then we go to metaphase 1. Those paired homologous chromosomes are all going to line up in the middle of the cell. So they're all going to form basically, the cell is like a round cell, and then right smack in the middle, they're all going to make a line all the way across. Once that, anytime you see a picture with cells all lined up, you know that you've already passed prophase and you're now in metaphase. So in prophase is when that nucleus starts to come apart and the pairs start to look for each other. 1 and 1 find each other, 2 and 2, 3 and 3, 4 and 4, like that. Okay, so the nucleus kind of breaking apart, that's prophase. And then when they all line up in the middle, that's metaphase. You can distinctively see, different phases or different pictures, if you will, of these phases, and you need to be able to identify what phase is going on here based on just an illustration. Once they line up across the center of the cell, Then we go to anaphase. And remember, we're still in the first part. Then we go to anaphase. Now in anaphase, these spindle fibers form. These spindle fibers look like these threads that reach from one end of the cell to the other end of the cell. Kind of like from the North Pole to the South Pole. And they reach all the way across. And when they do that, remember that section of the chromosome that stayed attached in the middle? That spindle fiber will attach to it. So you'll see these strings running north and south. And these split chromosomes, which we call chromatids at this point, are going to be attached to these strings in the middle, which we call the centromere, where they're going to be attached at that point. And remember, they're all still lined up in the middle, but now they have these fibers that run across them. And when that happens, then we're ready for them to start moving apart. So those threads are going to pull each homologous chromosome pair to opposite ends. So one is going to go up, one is going to go down. They're still attached in the middle. But remember those homologous pairs, 1 and 1, that sat here? So now those spindle fibers are going to pull this one up to the top, this one down to the bottom. So what happened? You had a diploid cell with 46 chromosomes, 23 pairs. Now, of those 23 pairs, each pair has done what? Moved to opposite ends. Okay? But there, in each pair also, each chromosome is what? Is split, cut in half on each end, but still attached in the middle. Okay? Now we go to telophase. Okay, remember, anaphase is where the homologous pairs are going to be split apart. And one is going to go this way, one is going to go that way. Once that occurs, telophase is basically going to make the nuclear membrane in the middle. The cell is going to basically elongate a little bit. And I think if I draw this out, you might understand just a little bit more. The cell, which normally starts out here, is going to have some of these on this side and some of these on this side. What I was referring to earlier as having that spindle fiber, I'll draw it on the bottom, is when the cell's like this, all these basically line up here across the middle. And the spindle fibers that I was referring to are basically these really thin strings that run along this way. And they attach right here, if this were the, they attach right here in the part right there. Okay? So it's a string that basically comes across this way. Now, there's going to be one there. There's going to be one right there. This one is going to go up. And this one is going to go down. So this line is going to basically split in half. These guys are all going to go up. All these guys are basically going to come down. When they do that during telophase, this cell is going to basically look like this. It elongates. And all these guys right here are going to bundle up in a small ball right here. A nucleus. And these guys right here that went down are going to do the same thing. And what are they going to do? They're going to bundle up and almost form a nucleus. You have a cell with two nucleus. Can you have that? No. So what's going to happen? This is going to move inward, and this nuclear membrane is going to move inward. And what you're going to end up with is something like this. They will detach right there. It'll just basically split apart. Okay? And now you form two new cells out of the one. You had one, and that one, when it goes through all this, it ends up with 1, 2. But you're still not done. You're still not done at this point, okay? Because now this same thing has to happen with an additional step. It has to happen all over again. So now, after telophase 1 is completed, now we go through meiosis 2. We go back to prophase. And now at this point, remember that each cell still had this chromosome split on the two ends, but attached in the middle. Now, during these 4 phases is where that detachment in the middle basically pull it apart. Okay? Now think about, go back to my analogy of the hot dog. If I took the hot dog and I cut it on this side, and I ended up splitting the 2 halves eventually, would I find any ingredients in one side of that hot dog that I would not find in the other? No. Think of chromosomes as the same thing. So genetically speaking, both of those halves are still going to contain the same DNA, the same genetic material. Now that's a problem, because if that doesn't correct itself during these 4 phases here, then guess what? Every single cell, every single haploid cell, reproductive cell, sex cell, that you would produce, you would always produce what? The same identical offspring. You would have a child to look like this, and 3 years later when you would have another child, guess what? It would probably come out the same, or the possibility would be there that it would come out the same. You could have twins 5 years apart. Okay, so obviously that can happen. So nature, if you will, has designed a way, or rather the body has designed a way to prevent that from happening. And that's the part where we talk about what's called crossover. Now in prophase, those chromosomes, each consisting of 2 chromatids. Remember how I told you that split? Each of those little leg type structures, if you will, when it splits like this part and this part, those are called chromatids. Okay, each one contains 2 chromatids. Now they become a little more visible. Okay, and then the final 4 phases of meiosis 2 is where we're going to turn that one cell that turned into 2, we're going to make those 2, each of them turn into another one as well. So 1 is going to make 4 in the end. Okay? So, meiosis is that process in which the number of chromosomes per cell is going to be basically cut in half. And you're going to separate those homologous pairs in a diploid cell to make haploid cell. Remember that there is a meiosis 1 and there is a meiosis 2. At the end of meiosis 2, that diploid cell became 4 haploid cells. All right, here are the pictures so that you get a better understanding of this. So we go back to meiosis 1. The cell undergoes this change where these chromosomes are going to replicate. It's also known as interphase 1. Now, each replicated chromosome is going to consist of those 2 identical chromatids, remember the little splits, and they're going to be joined in the middle. So again, just to make sure that we're clear on that, here you have what started out as this right here. You have your split right there, your split right there. You end up with this. This are what we refer to as chromatids. Where it's attached in the middle, that's called the centromere. But right now we only want to concern ourselves with this. These are called chromatids. Okay? And they're joined in the middle. Remember that this side right here is identical to this side right there. But when they split right there, this side is going to go up, this one is going to go down. I think sometimes I convolute my drawing so much that now you can't even tell anymore what's there. All right, so you have your chromosomes consisting of two identical chromatids still joined in the middle. And now the cell begins to divide those chromosome pairs. So what's going to happen, as you can tell, that cell has already started to elongate just a little bit. Now, the chromosome pairs are basically going to split. See the split right here? This is chromosome number 1. Chromosome number 1, they find their homologous pairs. All right, and then they're going to split. The whole entire thing, once it has split and stayed attached in the middle, it's called a tetrad. All right, so once it's as one chromosome, we just call it a chromosome. Once the split occurs in the first part of meiosis or of prophase, now it's called a tetrad. It's the same thing, we just give it a different name. Okay? Throughout all these phases, you're going to see that, where it's basically the same thing, but its name has now changed. Why do we do that? Because if I give you a specific name, you can tell me exactly in what phase this object that we're talking about is in. You get it? Okay. So now we form a tetrad when the split occurs. All right, it contains 4 identical chromatids or chromatids. 1, 2, 3, 4. Once that happens, they're going to start to pair up. Remember, before they go up to the middle, they're going to pair up. This is very important that you understand that this happens in prophase 1. Okay? In prophase 1, we go through the process called crossing over. Crossing over is very simple. See how this one is all red, this one is all yellow? What they're going to do is they're going to line up together. Remember when I drew my circle with all the X's, one on the top, one on the bottom? They're going to get really close together, and they're actually going to cross one over the other. And when they cross one over the other, they're going to exchange genetic material. Think of it as like two people, one going over and standing right next to the other. And then they exchange their left shoe only, and maybe a shirt and a watch or earrings or jewelry or something. Now they're still the same, but they've exchanged a little bit of what once belonged to them. One shoe, one article of clothing, maybe glasses or a hat, something like that. Okay? By that little change occurring, guess what? Now the genetic material is slightly different in both of them. So the picture color codes this for you to better understand. This is still that same chromosome, but it's no longer entirely red. It has what? It has a little bit of yellow. This one, after crossover, it's not all entirely yellow. It has that little bit of red. This is what prevents all the genes to be identically the same once these cells split and go their separate ways. So in haploid cells, those haploid cells will not be identical to each other, and nor will any of the 4 be identical to the way they were when they were just one. Okay, this is to ensure that when we pass on to our offspring, we're not passing on the same genetic material each time. Okay, so now, once crossover occurs, then they're all basically going to produce new combinations of the same alleles in that cell. Now at this point, they're ready for metaphase. And in metaphase, remember what I told you, what happens during metaphase? They all do what? They all line up, and these in the middle of the cell, and these spindle fibers start to form from one side to the other, attaching right here in the middle. Okay? Now remember, see how the color codes of these chromosomes show that they're no longer identical to what they were before this all started. So the homologous pairs during metaphase 1, the homologous pairs are going to line up in the middle of the cell, and they're basically side by side. The spindle fibers attach in the centromere, in the center part, and then they're ready for anaphase. In anaphase, the spindle fibers are basically going to pull them apart. So this one is going to come up, this one is going to come up to this side, this one is going to come down to this side. Okay? When anaphase 1 is complete, the separated chromosomes are basically going to just cluster around on opposite ends of the cell. Okay? And then we're ready for telophase 1. In telophase 1, you're going to get a small nuclear membrane to form around each of those clusters. Cytokinesis follows telophase 1. Cytokinesis is basically when this right here and this right here, when the outside parts of the cell, see how they kind of pinch in a little bit? Trying to get the cursor to show here, but now it doesn't want to. When this right here and this right here basically start to move inward. So what's going to end up happening? When the cell elongated, this is going to come up here, this one is going to go over here, and this point are going to come in, pinch together, and they're going to fuse. That membrane is going to fuse to form two separate cells, two new cells. Once that happens, meiosis 1 is done. And then we go to having two separate cells. And now we're ready for meiosis 2. And in meiosis 2, basically the same thing minus the crossover portion. In meiosis 2, the same thing happens all over again. Remember how this chromosome never split apart? Well, now in that middle part, the centromere, is going to be when it's going to split apart. So meiosis 1 is going to end up giving you two what we call daughter cells. We always use that term, daughter cells. Parent cells, that one diploid parent cell is going to produce two daughter cells. Okay? Now, each pair of homologous chromosomes were separated. That means that neither one of these two cells have a complete set of chromosomes like they did when they were in a diploid cell. Remember that a diploid cell was what? 23 pairs. Now each pair has gone off in its own direction. So there's only 23 here and 23 there. Okay? So they're not complete anymore. Now these two cells produced in meiosis 1 are going to have these sets of chromosomes. So now the final portion is going to be where this centromere needs to split so that this one produces 2 and this one produces 2. So in the first, the two new cells are going to go through meiosis 2. Now, unlike the first division, neither one is going to go through a round of replication. Okay, remember, crossover only occurs in meiosis 1. So what happens? Prophase 2. Basically the same thing. In prophase 2, their chromosomes, each consisting of 2 chromatids, are basically going to come out and become visible. The chromosomes don't pair up to form tetrads because the homologous pairs were already separated. There is no homologous pair at this point. So all they're going to do is basically in prophase 2, they're going to become visible. And then in metaphase 2, they're all going to basically line up in the center. Again, the spindle fibers are going to form, and you're going to have a line of all 23 of these chromatids or chromosomes, if you will, chromatid pairs. All 23 of them are going to line up. Spindle fibers are going to attach right in the middle, and that is metaphase 2. In anaphase 2, now is when that split is going to occur. So where they were split in the middle, look what happens. They basically split apart. Half goes to one side, and the other half goes to the other. Both actions occurring simultaneously in both cells. Okay, what happens in this one is also happening at the same time and in the same manner in the second cell. So when the cell enters anaphase, those chromosomes are going to basically split apart. Half go to one side, half go to the other. And then again, just like in telophase, basically the cell is going to kind of pinch together in the middle. When these guys form to opposite ends, the cell is going to pinch in the middle. A nuclear membrane is going to form around this group, another one around this group. You have a cell with two nucleus, or two nuclei, however you want to pronounce it. So, you can't have that. So, this is going to pinch together in the middle. This membrane is going to fuse together. This one is going to end up on this side. This one is going to end up on this side. Same thing over here. It's going to pinch right here. This one is going to end up on this side. This nuclei is going to end up on this side. And this one becomes 2, and this one becomes 2. Altogether you have 4. So these 4 daughter cells now contain the haploid number of chromosome set that you ended up with in the beginning. When telophase 2 is complete and cytokinesis is complete, you end up with 4 haploid cells out of 1 diploid cell. Okay? And that is meiosis.